In your postdoc PI, Steve George.
And Greg Longmore.
I'm gonna forget Greg because, or I could pull this up. Do a proper
Do my CV.
introduction.
I could send you my CV real fast.
It doesn't really matter after PhD. You know, everything else is just.
Dave Merriman's lab is the most important thing that ever happened.
Steve
Right?
George and
Greg Longmore.
Greg Longmore.
Yeah. Longmore. And it was actually split between biomedical engineering and molecular oncology. If you want to get that detailed. You don't have to.
You know what? They just asked me literally like 30 seconds ago, so.
I can't believe you don't know my background.
I know your background.
I can introduce you.
Of course I know your background. But you know how like I've been in seminars where people are like very eloquent in their introductions, and I kind of feel like it's going to be whatever comes out of my brain.
Do you remember Dave once saying that if you could put your people's skills together with my presentation skills, it would be perfect?
Yeah. I do remember that.
I think about that a lot.
I remember that because Dave made me feel like I could not speak publicly.
And he made me feel like I was just a terrible human being. So, you know.
So you gotta love Dave. Okay.
I'm gonna slightly turn this guy to help me. Because I really do like to walk around.
You ready? Okay. All right, let's get started. It's my pleasure to introduce Dr. M.K. Sewell-Loftin to Louisville and also to our seminar today. Dr. Sewell-Loftin is a great friend and colleague of mine. We've known each other for many years. She got her undergrad and master's degree at the University of Alabama. And then she took her talents to Nashville, Tennessee, to Vanderbilt University, where she worked with Dave Merriman and also myself. We were lab mates in Nashville. And after she got her PhD, she went to WashU St. Louis, worked with Steve George and also Greg Longmore.
There you go.
Yeah. And so after her postdoc, she started a faculty position in January 2020 at the University of Alabama in Birmingham. And her research centers around the cancer microenvironment using bioengineering tools and also to dissect the mechanobiology of the tumor microenvironment. So we're excited to have her here, and let's thank our speaker.
Well, thank you. I'm really excited to be here. I've spent a lot of time in Louisville, but I've never actually been to the university before, so this has been a really fun trip for me. And I hate that I kind of only had 24 hours and a really brief window, but I'm very, very excited to get to speak to you today about my presentation, about some of the exciting data that's coming out of our lab. And it's literally coming out of our lab right now. We had a paper accepted last week, well, provisionally accepted last week. Mackenzie Johnson's actually an author on it. And we have another one that's finishing up review in scientific reports. And so they both should be out soon. So all of this is literally brand new, exciting things that have not been seen before. So like Dr. Chen was saying, I really focus on this idea of mechanosignaling inside the tumor microenvironment. And we're interested in these mechanical forces and the way those forces interact with cells and tissues to actually alter not only disease progression, but the overall response to chemotherapies. And I don't think I need to spend a lot of time telling you why cancer is bad. Cancer is obviously the second leading cause of death across the entire world. And while we have gotten significantly better at diagnosing it at earlier and earlier stages, you can see that our mortality rates haven't necessarily improved a lot over the last 40 to 50 years. And 90% of all cancer deaths are related to metastatic spread, or when the tumor leaves its primary site, goes and travels throughout the body to lodge in the secondary site, and then that secondary tumor growth is really what winds up killing a lot of people. And that's because these metastases are usually more resistant to the chemotherapy than the primary tumor was. And so when I first started working in Dave Merriman's lab with Joe, I was really interested in this idea that mechanical forces change cell behaviors. And then when I went to do my postdoc and I moved into the cancer environment, I wanted to translate that into what mechanical forces drive tumor growth. And I first landed on a project related to angiogenesis. And so in 1971, I believe, a paper came out that basically said that without angiogenesis, tumors won't grow whatsoever. And this launched an entire wave of therapeutics around blocking angiogenic growth. specifically vascular endothelial growth factor, VEGF, or its receptor, conveniently named VEGF receptor. The reason angiogenesis is so important is that as a tumor mass increases in size, what happens is it will eventually reach a threshold where it can no longer receive sufficient oxygen or glucose through passive diffusion. And that's about 200 microns in diameter, actually, so it's pretty small. And the tumor will create a switch for pro-angiogenesis or pro-vascular growth. And so it will make nearby capillaries actually begin to sprout new vessels. And these vessels will grow into the tumor. And this allows two main things. The first is that the tumor can now receive the nutrients that it needs, so it can get even bigger. And the second is that it provides a pathway into the vasculature for metastasis to occur. So again, the theory was if we can shut down the signaling that drives angiogenesis, can we prevent tumor growth? And the answer has been kind of, honestly, from a clinical standpoint. And I'll talk about that more in a minute. But when I first saw this, what really fascinated me is this actual sprouting of the capillary to grow this new vasculature. This is very similar to processes that naturally happen during embryonic development, which had been what I studied as a PhD student. And so I actually wanted to investigate this first wave of angiogenesis. And this occurs when you have some kind of quiescent vessel that receives some kind of stimulus to create what we call a tip cell, shown in green in this particular figure. And this tip cell becomes highly migratory, and it becomes able to interact with the matrix more than its cell-cell neighbors. And so it begins to move and remodel the matrix. It begins to actually pull these purple stalk cells forward to create this branching vessel. Now, the biology that drives that is actually very similar to epithelial to mesenchymal transformation, or EMT. And we know that interstitial flow can do this. That's been shown forever in a bunch of tumor models. We actually think it might be related to integrin activation now. And we know that soluble growth factors, things like VEGF and HIF, which is hypoxia-inducible factor, can make that happen. But my PhD work and some work out of Celeste Nelson's lab at about the same time actually showed that strain can induce EMT-like shifts in various cells. And so my question was, do mechanical forces in the TME actually create a pro-angiogenic environment independent of this interstitial flow and independent of these soluble factors? And that was really the launching point for most of the work I'm going to talk about today. What kinds of forces are present in the TME, and how do these forces drive not just angiogenesis, but also tumor growth in general? And to answer that question, you first have to say, well, what's going on in the tumor microenvironment that can actually make microstrains that might be causing this? And I started focusing on cancer-associated fibroblasts. So tumors are not just tumor cells. Our lives would be so much easier if they were, because it'd be a single type of cell that we're trying to deal with. What actually happens in the TME is you have cancer cells, you have fibroblasts, you have mesenchymal stem cells, you have immune cells, you have the endothelial cells of the blood vessels. You can see where I'm going with this. This is very complicated. But cancer-associated fibroblasts actually usually arise from quiescent resident fibroblasts in the adjacent tissues. And when it is quiescent, it's just a nice little fibroblast doing homeostasis events. When it becomes a calf, it becomes an activated phenotype. So this is more myofibroblastic. It has increases in alpha smooth muscle actin, SNAIL1, and it dramatically increases the amount of collagen secretion and reorganization that is happening in the area around it. The increase in alpha smooth muscle actin helps drive increased contractility of these cells. And that has been shown in 2D. This is work from Cindy Reinhardt-King's lab, where she took normal fibroblasts and calves from breast cancer patients and measured traction forces, and she saw that basically these calves are much more contractile. They want to induce larger strains and larger forces in their substrates. This was also shown with some work out of Greg Longmore's lab when I was there, where these are calves that are either scrambled control line, or this set of calves has a collagen receptor knocked out. And so when the calves are no longer able to hold on to the matrix around them, they're no longer able to deform it, they can no longer generate traction forces. And this differentiates calves from their normal fibroblast counterparts. So I really wanted to be able to focus on this kind of mechanical activity, these contractility events on calves in the TME. And everything that I had read, everything that I had seen had all been in 2 dimensions, which is great. It's necessary models, and it helped us fundamentally understanding these cell behaviors. But I wanted to develop some higher level systems to ask some higher level questions. And I've developed this figure over several years. Parts of it are borrowed from Steve George, lovingly. And as we go from the left side of the screen to the right side of the screen, we can see our traditional cell culture and cancer models. So we have 2D cell culture that happens on tissue culture plastic, which looks like absolutely nothing that is physically present anywhere in our bodies. We can do 2.5D or 3D cold culture systems. Mice are currently considered the gold standard at the NIH for doing preclinical cancer models, but mice are really terrible people. This can be with immune cells or without immune cells, but all of the mechanical properties in mice are very different than in humans. So eventually, obviously, we want anything we make to make it into a human patient. The problem with this is while you are increasing complexity, moving left to right, you are also dramatically increasing the amount of time and money it takes and the safety involved with making sure that these experiments are performed ethically and robustly. As I've developed this figure, what I've actually decided on is that we can take advantage of this opposite trend, where we have more control as we go into these simpler models. And so where we have landed is this kind of ex vivo co-culture niche, where I can independently leverage some control features that aren't present in mice, but that still provides me with more complexity than what you would find in traditional 2D cell culture. And so this is actually a really pretty picture out of Steve's lab. I should probably switch it out for a really pretty picture from my lab, but it's doing the job right now. So the first part of this talk is going to be on the platforms that I developed to really investigate these mechanical behaviors of calves and then tumor progression. And that started with saying, well, are they really more contractile in 3D? Do we really see differences in calves compared to normal fibroblasts in the 3-dimensional models? And so NBF stands for normal breast fibroblast. These are breast cancer fibroblasts from the same patient. And what we have here are vector maps that are tracing the movement of 1-micron fluorescent beads in a 3D gel. And I realize this now says E formation, that should say deformation. But essentially, these are matrix distortions that are generated by a contractile event of fibroblasts that have been embedded in this gel and tracked over the course of up to an hour. We've done 20, 30 minutes, up to an hour, and we see similar behaviors kind of at both time points. And this is poor man's traction force microscopy. So if you've ever seen traction force microscopy, it's really pretty. It's like a single cell, and you can measure individual movements of individual pieces of the cell. This is more of a voxel. This is about a 50 micron by 50 micron by 75 to 100 micron volume of gel that we're looking at. We take all of those data points and we can feed them into various computational models and algorithms, and we can come up with average matrix distortion magnitude generated by these cells. So black is our NBF, red is our CAF, and what we see pretty extensively is that CAFs are always going to generate larger matrix distortions compared to NBFs, larger micro-level strains inside a matrix. This was really cool, but CAFs also secrete a bunch of growth factors. They put out VEGF, they put out HIF, they put out stromal-derived factor. So I needed to come up with a way where we could make these matrix distortions happen in the absence of stromal-secreted growth factors. That led me to develop a magnetic bead assay. So we take iron oxide magnetic nanoparticles. They have a tosyl activity on the outside of them, so we can coat them in thrombin. If you mix those then into a fibrin-based gel, what happens is you wind up with magnetic beads that are effectively but not chemically cross-linked into this gel. We can then load in our 1-micron blue tracer beads as well, and if we put a magnet next to it, what happens is everything moves together. And I did some microscopy. This is from a Scientific Reports paper in 2017, where I kind of showed that the movement of the magnetic beads corresponded exactly to direction and magnitude to the movement of the blue tracer beads in the system. And so this way we can introduce stromal-like deformations, stromal-like mechanical activity, without having growth factors. So now we're getting to the ability to tease out mechanical from biochemical cues inside a 3D gel like this. To prove that it actually worked, I took these magnetic beads and I put them into gels with endothelial cells only. Endothelial cells like to make blood vessels, but they won't do it without some extra growth factors or stromal support factors. And what we actually found is with endothelial cells only and these magnetic beads, it could dramatically increase the amount of vessels that grew in the system. I then went through and I had some genetically modified lines, so NBFs don't normally make vessels, calves do. And then this is a ROC2 knockdown or a YAP knockdown. And basically, mechanical stimulation rescues vessel growth in stromal populations that normally would not support vascular growth. Very exciting, published this paper, and Steve George looked at me and said, "Congratulations, you've made blood vessels, but they don't have any flow in them." And he was right. There was no flow in these blood vessels because this was a static system. So I started working on a microfluidic model so that we could create blood vessel networks and control not only interstitial flow around them, but also grow fully connected in anastomosed vasculature. This is the workhorse of the lab. We call it the centipede device because it's long and spindly and it has a lot of little processes sticking off of it when you really look at it. And the blue regions are tissue chambers. So anything that's blue, I can put in cell matrix, I can put in different cell populations, I can load them on different days at different times. And they're connected by 20 micron ports at these interfaces. The pink parts of this figure are the fluidic lines. And because we have fluidic lines on each chamber independently, what happens is I can now control flow in both the y-axis vertically and the x-axis horizontally, which allows me to control convective diffusion of molecules between chambers. So in this way, I like to tell people that we have independent knobs in this system. So I have this complexity level, but also a control level. So I can change interstitial flow, I can change BCM composition and stiffness, and cell phenotype. So that actually worked the way I told Steve that it worked. He asked me to do some COMSOL modeling. And essentially, I filled all of the chambers with fibrin gels. In this particular graph, positive velocity means that flow is moving from left to right. And so in this model, what we actually see is there's very, very little flow at these interfaces between chambers. So this is microns per second. And you can see these red circles are where those chambers happen, that chamber interface happens. And there's virtually no convective diffusion. Cartoon-wise, it looks something like this. Also created an outward flow pattern. So now we put high pressure in the center chamber, low pressure in the side chambers. And what we do is we create these peaks where this is moving from the center to the left, and this is moving from the center to the right. And anything that was loaded originally in those side chambers can now not diffuse against that flow gradient to make it to the cells in the center chamber. So this is effectively eliminating a biochemical stimulus from the cells in the center chamber at that interface. It looks like this. And then I decided to actually do it in real life. So this is FITC and rhodamine tagged dextrin that's been loaded into this device. This is that top to bottom flow. So there's no, very, very little diffusion of these interfaces, which you can see in this graph here. And then I take this exact same device and I put it in the outward flow, and we see everything is flushed out. And now, even though I put a ton of FITC in these side chambers, there's not significant FITC making it into the center chamber. To prove that I could keep cells alive in this, I took normal fibroblasts, normal lung fibroblasts, which make blood vessels and endothelial cells, and loaded them into the center chamber. And then I used either our CAFs or our mechanically inhibited CAFs in the side chambers. And then we also made this fun line of a constitutively active row added to the normal fibroblasts. And what this did was it created a normal fibroblast line that had enhanced contractility. And we tracked that vessel length was still associated with the mechanical activity of these stromal cells, even when the flow is outward. So anything that's secreted here is being washed out to the left in big secreted here is washed out to the right, do our blue tracer bead protocol in these devices as well. So it can actually look at mechanical interactions of cells, matrix deformations generated inside these communication ports between chambers. And what we see is our cath EVs make matrix distortions, SHF knocks them down, and our NBFCA row actually has increased matrix distortion compared to both our knockdown cells and to our NBF EV cells here. So all of this is, and this is lab on a chip. This came out a couple of years ago. And this was basically showing that, yes, we generated this platform. We can track mechanical differences. We can track functional differences in cells. And when I started my faculty position, the question was, how does this actually matter for tumor growth? Because as you may have noticed, I haven't talked about a single cancer cell in any of the data I've presented so far. All of this was cancer agnostic, intentionally to help me build up some collaborations. But now that I've moved into my own lab, I'm starting to actually set up some specific cancer projects, and a big one we have is an ovarian cancer project. Ovarian cancer has about a 50% survival rate for 5 years. And this is largely due to recurrent and chemo-resistant disease. But a part of it is also due to late-stage diagnosis. The earliest signs of ovarian cancer are a little bit of abdominal bloating and some extra nausea. So a lot of individuals will show up at their doctor's office and say, hey, I'm feeling a little nauseous, a little queasy, a little bloated. And the doctor tells them they probably have IBS and sends them home. And several years later, they have metastatic ovarian cancer. And one of the reasons ovarian cancer is slightly different from a mechanical standpoint is ascites fluid buildup in the abdominal cavity actually is a huge indicator of disease. And there are patients who develop 3 and 4 liters worth of additional fluid that have to be drained. And so this fluid buildup creates extra compression. It also is more fluid to create shear flow and shear stresses on the cells in the area. And that's how metastasis actually occurs for ovarian cancer. That acidic fluid will circulate through the cavities. As it does that, it will shear cells off of the ovaries and fallopian tubes. They will move around in this fluid and land on either the omentum or the peritoneum, and they will begin to invade through those layers and into the rest of the abdominal cavity. That's kind of the macro mechanical forces. We also still have those micromechanical forces. So if we zoom in, we have our tumor growing here. We have fibroblasts that want to secrete extra collagen and remodel it and make it stiffer. That creates a tension at the periphery as well as compressive forces on the inside of the tumor. That acidic fluid flow is still creating shear on the outside of this. And if we go in even further, we still have the individual forces generated by these cells too. So our cancer-associated fibroblasts are a source of tension even as the tumor is moving. We have the compression from the acidic fluid that's affecting individual cells. And then the interstitial fluid flow also can affect these cells. So this is a very complicated mechanical environment. And our mouse models for cancer, for ovarian cancer specifically, are pretty terrible. It's hard to get the tumors into mouse ovaries because they have some additional features that human ovaries don't have. And it's also one of those things where the average age of diagnosis for somebody for ovarian cancer is 63 years old, which tends to correlate with postmenopausal cycles. And old mice cost a lot of money. So it is very expensive to use a mouse that accurately mimics the aged population of ovarian cancer patients. The other thing I just want to mention on this slide really quickly is some of the commonly used chemotherapies. Paclitaxel and cisplatin. These are used for more than just ovarian cancer. They're also used in breast cancer, lung cancer, gastric cancers. And paclitaxel targets microtubules to inhibit DNA synthesis. And cisplatin actually cross-links urine bases inside DNA, also to prevent DNA synthesis. I want to point out that these are 2 clinically widely used molecules. None of them do anything about mechanical features of the tumor. They all target DNA. They all target proteins and receptors. And recently, we've really noticed that cisplatin and paclitaxel also have a large range of off-target and post-target effects that involve mechanotransductive elements. So when you give a patient paclitaxel or cisplatin, or you give cells and culture these drugs, what you will see is you will see changes in Rho-ROCK contractility proteins. You will see changes in NAP, nuclear orientation localization, even though that's not really what they're targeting. So how can we actually study the mechanosensitivity that's driven by mechanical stimulation in ovarian cancer cells? I have two pictures here. This is Molly Buckley. She was an adopted PhD student in my lab, and I'll be happy to tell that story later. And Miranda Tidwell, who was the first PhD student I hired into my lab. And they came up with this system where we put endothelial cells and normal fibroblasts to make blood vessel network here. And then we put ovarian cancer cells in the left and the right chamber of this device. And we did the outward fluid flow. So there's only media in the center fluidic lines. We used a couple of models of epithelial ovarian cancer, OVCAR8 and SCOV3. These are both Ceres grade, although there's some debate about where they actually came from because they've been immortalized cell lines now for 30 or 40 years, so maybe it's been some drift. But this was all based on the fact that Molly's first paper in her PhD had taken these cells and put them on a flex cell plate for 24 hours or up to 72 hours. Then she harvested those cells and injected them into SCID mice, so immunocompromised mice, because these are human ovarian cancer cells. And what Molly saw is that you actually got increased metastatic growth in cells that had been strained for longer times compared to non-strained controls. And then she went on and they did some RNA-seq on these cells, and they actually found a whole lot of the EMT markers are upregulated. The mitten was upregulated, cadherin 11 was upregulated, snail was, I think, up 3 or 4-fold, and things like E-cadherin were significantly downregulated. So that paper was published literally right as I started at UAB, and Molly said she wanted to figure out why that was happening, and that's when she designed this experiment. And as we were looking at things in the setup, she came across heat shock protein 27, or HSP27. She dug back through her RNA-seq data, and the gene that regulates HSP27 is HSV1. It's actually increased in cells that have received strain. It's also been identified as one of these key post-target effectors of cisplatin and paclitaxel. Basically, you get more HSP27 when you give these drugs. And another group had recently shown that it was sensitive to shear stress. In other words, if you put cells on a 2D plate and you put flow over the top of them, phosphorylated or activated HSP27 wants to move to the forefront of where that shear is inside the cells. So we thought maybe this would be something to look at to figure out if HSP27 is altered in strain in our models. So the first thing we did was we took a flex cell, which for those of you who are not familiar with that, it's a 6-well plate that's got a silicone membrane on the bottom instead of plastic. You put that on top of a base plate in an incubator. That base plate is attached to a vacuum pump, and you can control how much vacuum is initiated. So you can essentially introduce very controlled cyclic or static strains in these cells. So we did cyclic strains for, well, no strain control, 5, 15, 30 minutes, and we looked at phosphorylation of HSP27, total HSP27, and then here's our beta active floating control. This first slide I'm showing is the SCOV3 cells. But we also had a derived line from these SCOV3s called .TR. And essentially, if you give SCOV3 a really low dose of paclitaxel and just kind of keep them in that, eventually you wind up selecting for a Taxol-resistant subpopulation of these SCOV3 cells. And so we didn't see a lot of changes necessarily in our SCOV3 line, but in this TR line, we saw large increases in phospho levels or activated HSP27 levels, particularly in 5 and 15 minutes. And then this is just the quantification of those plots. And the big things that I want to take away from this data is that the TR lines have more phospho HSP27, regardless of strain treatment. It just seems to be upregulated in these cells. But a parental SCOV3 line, going from no strain to 5 minutes to 15 minutes, dramatically increases our total HSP27 expression. And we don't see that in the Taxol-resistant cells. I'm going to talk a little more about how this all fits together as we go through some more data, because I know it was kind of a head scratcher when they first brought it to me that we'd see different behaviors in these different cell lines. I will say this is the paper that's hopefully finishing up review right now in Scientific Reports. So this is not published yet, but the first round of revisions was pretty positive, and we feel good about it. So we then discovered that there were ways to inhibit HSP27 using an available drug on the market. It's called J2, some small organic compound, and we gave the SCOV3 and TR cells the J2 overnight before we put them on the flex cell for 15 minutes of strain. So this should have increased our HSP27, particularly in our parental line. And what we actually saw is we don't get that strain-induced increase in total HSP27 when we have this J2 inhibitor. So this soluble inhibitor is blocking a mechanically induced signaling event. It's going to be really important for later in the talk, so hold on to that little nugget. And then we said, okay, let's put this in devices, because the flex cell is great, but it's still all two-dimensional. So our timeline is going to be down here in the left-hand corner, and we do either 72 or 24 hours of pre-treatment with strain before devices are loaded on day 0. For days 1 through 7, the device will get fresh media and that outward flow pattern to wash anything secreted in the side chambers outward. And on day 8, they get fixed, and we stained for HSP27, cleaved caspase 3, which is a marker of apoptosis, and DAPI as a nuclear counterstain. And so here's our device setup again. You can see we've got tons of cells in the center. Here's control non-strained cancer cells and strained cancer cells. And then this is just a blown up image of this. This is our 24-hour picture. Our 72-hour pictures look a little different. You can tell maybe the cells didn't quite grow as much as we wanted, which is another avenue that we're now trying to figure out is proliferation. But we ran these side chamber images through ImageJ to normalize our HSP27 expression levels to total cellular count. And what we actually see is that strain still dramatically introduces higher levels of HSP27 in these SCOV3 cells at both 24 and 72 hours, although the levels do seem to drop off with longer strain treatments. Now, what's interesting about this is you have to remember these have been in culture for 8 days beyond the strain treatment. So this is something that is retained as a mechanical memory inside of these SCOV3 cells, that there is something that is still creating this HSP27 upregulation. So again, strain increases HSP27 after 8 days in particular, and longer strain treatment seems to decrease this effect. The next check was to actually look and see if we could get paclitaxel into this and monitor cell death. So now it's the same timeline down here, except on day 4, we're giving paclitaxel to the devices. Now this paclitaxel is going into that center chamber, so it's going into the normal region and is then diffusing and flowing out to those side chambers where the cancer cells are. The reason this timing matters is it allows for a couple of things. First, we trypsinize the cells, and it's kind of traumatic for them, so we want them to reacclimate and actually be seeded into the 3D model. And second of all, this might be a little more relevant clinically than just putting cells down and giving them the drug immediately, because your cancer's going to grow for a little bit before they figure out what they want to treat you with, and then they're going to wait a little bit longer to see if it actually took, to see if it worked. So now we're treating on day 4, fixing on day 8 again, and now we have our HSP27 in red, which is increased in our strained TR cells at 24 hours in particular, and our cleaved caspase 3, which is in green, which is decreased with longer strain treatments. So the important parts of this information is that now we're seeing this increase of strain in 24 hours in our TR cells after they've been treated with this paclitaxel, but our apoptotic markers don't increase, even with paclitaxel treatment, even without strain. Now, cleaved caspase 3 is an early apoptosis marker, so these are cells that have been identified to undergo apoptosis but aren't truly dead yet. Because of some of our fluorescent reporters used in other studies, we couldn't do just like a straight up viability assay in this particular model. But the paper does have some cytox data for these cells that have been treated with strain and treated with paclitaxel. And it's the same kinds of things. Decreases in apoptosis and in overall cell death in strained TR and strained SCO3s once they've been treated with the flex cell. We next did the experiment but included J2, so our HSP27 inhibitor. So again, the cells are cultured in that while they were strained. So we're inhibiting HSP27 expression during that strain treatment. They grow for 4 days in the devices. We treat them with a paclitaxel, and then we collect these images of HSP27. And what we actually see is, again, J2 blocks expression. It's always great when your inhibitor still inhibits things when you do a new experiment. But dramatically, our TR cells going from strained vehicle treatment to strained J2 treatment, we have again blocked this increase in HSP27 upregulation. So even though these cells were treated with strain early, were treated with J2 early, we are shutting down the stress protein response when we give these cells some kind of strain stimulus. We also tested this in that other cell line, the UofCAR8s, and we see very similar behaviors. We have increased HSP27 after 24 hours of strain, but not 72 hours. And at 72 hours, we see overall less cell death and less apoptosis markers in cells that have been strained compared to non-strained cells. The takeaway from this paper is that HSP27 can be increased due to tensile strain, specifically in these ovarian cancer cells, and that strain-induced activation of this molecule is blocked with a soluble inhibitor. That's very important from a druggable target standpoint. You cannot create a drug and give it to a cancer patient if it's not going to actually shut down the mechanism that's driving that signaling pathway, which is some of the data I'm going to show here in a second. So the fact that this soluble molecule blocked HSP27 expression and activation made us really excited, and we did see some decreases in apoptosis markers. The other takeaway from this is that biology is really complicated still, because these are 2 parental lines of epithelial ovarian cancer, 1 derived line from that, and we did see some divergent expression of HSP27. And what this could suggest is that the Taxol-resistant cell line, how it becomes Taxol-resistant, may not rely explicitly on our HSP27 signaling. So more work is needed on that, and we have some avenues of investigation we're starting to pursue, but we're excited to hopefully get this paper off, especially since Molly graduated in 2022, and she's ready for me to stop emailing her asking for Western blot analysis now. As Molly finished up, Miranda actually took a lot of this work and said, okay, that's great, but what about receptors that might actually be transducing these forces into cells? And I said, all right, go look at one. And so she first started with CD44. And we started with CD44 because it's easy to look at CD44. It's on a lot of different cancer types. It's associated with increases in EMT, which is related to metastasis. It's also associated with cancer-like stem cells, cancer stem-like cells, possibly with dormancy. And it's on both ovarian and breast cancer, which are the 2 main models we look at in my lab. And if you look at this figure from a lovely review paper that's a little older now, but still good. When HA, or hyaluronic acid, binds to CD44, it can interact with this anchoring construct to connect directly to the cytoskeleton. The intracellular domain of CD44 also interacts with the Rho-ROCK pathway. Again, suggesting that CD44 has some role in regulating the mechanoresponses of the cells. And this is even more interesting to us because hyaluronic acid, or HA, is both a ligand and a matrix cue. This is a large glycoprotein. This is a large biosugar, abundantly present in different tissues. And we know that it actually can increase in the ovarian stroma over time with patient age and with disease progression. We also know the places that breast cancer and ovarian cancer like to metastasize to contain a lot of hyaluronic acid, places like your bone marrow or brain. Those are very conducive environments for this growth. And so the question really was, is HA a mechanical signal Is it a biochemical signal, or is it both? And does it matter to these cells? And the reason we got to ask this question is because as the ovarian cancers shear off of that initial tumor source and they're moving around in that ascites fluid, you now have basically non-adhered cells that could bump into HA molecules, because there is serum HA as well. So that's where this project really originated. And this is a little bit of a teaser data, because Miranda is frantically doing about 7,000 Western blots right now as we speak. But she took the OVCAR8 cells and she did 2D and 3D studies. And she either gave them HA in the media, just mixed it straight up in the media as a soluble molecule, so that's SOL. She would coat tissue culture plastic with it, so that's this PLT. And in 3D, she would either use fibrin only with soluble HA or fibrin plus a little bit of HA inside the gel. And then she did Western blots on this. And what we see very reliably over and over again is that when we have this matrix-bound HA, whether it's on the plate or whether it's in the gel, we increase the amount of CD44 expression that's happening in these cells. And I think this study was maybe 2 or 3 days. I think this study is a week long. She's repeating the experiments now, but we see if we actually do this in a strain environment. So if we take this setup, but put it on the flex cell plates and we strain it, we see another 10 to 15% increase in CD44 that happens with a combination of matrix-bound HA plus strain. And so this is indicating to us that CD44 may be acting as some kind of mechanoreceptor or mechanosensor in a way that others haven't necessarily described before. So this is what Miranda's now building her entire dissertation around, and she's actually finishing up these experiments because she's going to propose here in the next couple of weeks. One of the other studies she wanted to look at was functional changes. And so she created a knockdown line, a CD44 knockdown line using shRNA. We have a scramble control, and we get about 70% overall knockdown. She treated these cells with or without strain on the flex cell, and then gave them a high dose of paclitaxel. And then because this was 2D, we could do our cytox staining. And that's, I apologize, it's not easy to see on the screen, but there are red dots that are dead cells and blue dots is the host live or total cell count staining. And when we look at the results, when we analyze this, we see very dramatic decreases in cell death when you strain these cells, as long as they express CD44. So wild type, so non-modified cells, here's our non-strained but with paclitaxel, and this is strained with paclitaxel. Non-strained with paclitaxel, strained with paclitaxel. And then that difference completely disappears in our SHCD44 cell line. And she has all of the permutations of with HA, without HA, with strain, without strain, 2D, 3D. She's got all of those, but the graphs get a little unwieldy. So I just wanted to show this one because I think this kind of encapsulates what's really happening very clearly, in that CD44 seems to be strain is protective against cell death as long as CD44 is expressed. So that's all of our ovarian cancer data. And now I'm going to switch gears a little bit. Talk about another project that's ongoing in our lab. This is the paper that actually got accepted last week. This was done by the second PhD student in the lab, Bronte Miller. McKinsey actually worked with Bronte for a year and a half, something like that. And essentially we went back to VEGFR2 and angiogenesis. The NCI had graciously given me a lot of money to answer this question, and so I wanted to make sure to do my best to answer that. We know that VEGFR2 is important. We also know that VEGFR2, despite it being a vascular endothelial growth factor, is aberrantly expressed on certain types of epithelial-based cancers, including breast cancer and ovarian cancer. If you go into something like the TCGA, you can actually screen for VEGFR2 gene expression, and there are certain subtypes of breast cancer that increased VEGFR2 expression correlates with decreases in overall survival. And that's cancer, not necessarily endothelial cells. And finally, we know that anti-VEGFR2 drugs are limited in their improvements clinically. The best example of this is bevacizumab, which was approved by the FDA in the early 2000s to treat breast cancer. It's been about 10 years being used clinically in combination with other chemo reagents. And effectively, there's no difference. There's no improvement in patients that receive an anti-VEGF drug plus chemo compared to chemo-only patients. So the FDA has actually since removed the approval for bevacizumab for breast cancer. Molecularly, that looks something like this. So this part in the middle is our dimer of VEGFR2, or KDR is its gene name. They can bind to its ligand VEGF. There are multiple members of the VEGF family. There are multiple members of the VEGFR2 family. But what happens is when VEGF binds, you get phosphorylation of specific tyrosine residues represented by these little P's here. And this is not an exhaustive list. And some of the pathways that it hits are, again, our good old mechanotransductive. This is the MAPK pathway related to cell proliferation migration. This is the SRC PI3K cell survival pathway. So these are known mechanoresponsive pathways. We know that strain alters their signaling. And they're downstream of VEGFR2. VEGFR2 also alters their signaling. So we wanted to investigate this in the context of mechanical sensing in endothelial cells to drive angiogenesis. And some of this was also based on work out of UCLA, Chen et al. And there's a lot of Western blots here, and I apologize. The important ones are circled for you, where these are different tyrosine residues that are phosphorylated. This is 1059, this is 1214. And they did VS, which is soluble VEGF, or VB, which is VEGF attached to the collagen matrix. So now it is a matrix-bound ligand, and they called it a mechanical stimulation. I might have to argue with them about whether it's a truly mechanical stimulation, but they said it was at least mechanical-like. And the important thing here is that at 15 minutes, all of the phosphorylation events are gone, no matter if it's soluble or bound VEGF, for every tyrosine residue except this 1214, which has a prolonged phosphorylation level at 15 minutes, but only with the matrix-bound ligand. I had found this paper when I was a postdoc. I showed it to Bronte on like day one. I said, hey, why don't you go put some cells on the flex cell and try to measure these residues and see if strain changes it. And she very graciously did not call me crazy, and she very graciously went into these Western blots. And so she took human microvascular endothelial cells. We did 10% strain elongation at 0.3 hertz, which is just a nice respiratory rate. And then she either did no treatment, so no VEGF, or she did a ton of exogenous VEGF. So we have NT, V, the epsilon is strain only, E plus V is strain plus VEGF. The left-hand side is at 1054, 1059, so the non-mechanically responsive residue, and the 1214 is the mechanically responsive residue. And what we see is that in these conditions, strain will increase our phosphorylation levels, and strain plus VEGF increases it even more for 1214. And at first I was a little sad that it was a both and not an either or, but then realized that this may actually be a better mimic for the tumor microenvironment. Because if we think about it, the tumor cells are secreting VEGF, the CAFs are secreting VEGF, and it has all of these altered mechanical properties. So this very well could be a both situation that's happening. We then took commercially available VEGFR2 blocker, SU5416, gave it to cells and did the same experiment over again. And now we saw kind of divergent behaviors where our 1054, 1059 saw some increases in phosphorylation levels with the inhibitor. And we didn't completely, abrogate our phosphorylation at 1214, even when this inhibitor was present. So this again goes back to the idea that there are these multiple inputs, this ligand input, this mechanical input, and when you provide an inhibitor like the SU5416, you might not be blocking all of that signaling. So this is in opposition to our HSP27 story, where I said we can give it a drug, we can shut down the mechanical activation. Hooray, that's a great targeting strategy. Here, when we give it a drug, we're not necessarily shutting down all of that receptor activation, which means that we need to come up with a better way to inhibit the mechanical activation of this receptor if we want to consider this a way to target blocking tumor growth. Auntie also did some really cool work in our 3D models, where she used magnetic beads and VEGF and this SU inhibitor. And the important thing on this graph is this mustard bar is not different than this mustard bar. which is going from our vehicle treatment with VEGF to VEGF plus our inhibitor. So again, this mimics the tumor microenvironment really well. And even when we give it mechanical stimulation, there's still, a slight increase in our overall vessel growth that happens. This was that static assay there with no flow. So Bronte also looked at this in our angiogenic assay. And so now we have our vascular network in the center, vehicle control, SU5416 drug, CAFs or NBFs. And what we still see is this orange bar, this inhibitor-treated bar, has significantly more angiogenesis that can occur than any of our normal breast fibroblast samples. And it's actually not different than our vehicle growth here. It's just a very large error bar. It was very, sometimes micro devices are like that. And secreted factors were washed out. So the flow is going from the center chamber outward. And so we know that this has to be driven by those mechanical features. Sure of that, we did our magnetic bead assay inside the microfluidic device. And we see the same thing. When we give magnetic stimulation to the system, we get increased angiogenesis without the addition of stromal secreted factors whatsoever. So we know that at least these matrix distortions that we can introduce are sufficient to drive some level of VEGFR2 activation, even in the absence of exogenous or stromal VEGF ligand. I can summarize now. I hope I've convinced you that we should definitely investigate and interrogate mechanical and biochemical signaling separately in the tumor microenvironment. This is going to be really important for identifying how mechanoreceptors can be targeted in new cancer treatment strategies. And I am starting to work towards this idea that mechanical forces actually regulate chemoresistance. And that might give us yet another avenue to figure out how to improve treatments for these patients who have recurrent or resistant disease. Finally, the lab has been, I think, very hopefully diligent at demonstrating that we can get differential activation and phosphorylation events for receptors and other stress proteins that haven't necessarily been identified before as strain receptors. Most people who talk about VEGFR2 talk about shear flow because it's in endothelial cells. We're the first ones really talking about this idea of tensile strain on the receptor. Then finally, I want to thank all of the people who helped make this happen. Obviously, our funding sources are critical right now. And then here's the list of graduates and undergraduates in the lab. I showed a little bit of Bronte's work. Michael's been helping her with that. Molly's picture's been taken out because she graduated, so she doesn't get to be in the group anymore. Same with Mackenzie. But Ali and Kamari have also worked on this project. We've got some more cool CD44 data from Adam that I wasn't able to present today, but I'd be happy to talk about later. And then just as, cover my basis, I do perform some consulting work for a company called Surflux. None of the stuff I talked about today is related to what they do though, so it should be in the clear. Thank you very much, and I'm now happy to take any questions. Yes.
I was just wondering, how are you applying the strain?
So we have both a flex cell system, which is one of our 2D substrates. So it's a silicone membrane, sits on a vacuum plate. You can just basically stretch them. We also have, let's see if I can get back to this without like too many button clicking. Go to slide number. Nope, just gonna make me. All right. Well, we have a magnetic bead system. There we go. And we can make a 3D fibrin gel, and we put magnetic beads inside of it that are coated in thrombin. So when the fibrin polymerizes around those beads, they're effectively cross-linked into place. And if you put a magnet just underneath that on an orbital shaker, those beads will move around. So that's a way to induce these matrix distortions of mechanical activity without having something like a fibroblast nearby. Good question. Hi.
I'm not really familiar with the large cell stuff, so apologize if
No worries.
It's kind of a simple question. You said your system that you made, I think the voxel size was up to 150 micron or something.
Yeah.
You stated that tumor cells that start generating these new angiogenesis essentially is about 250 microns. So is that size difference potentially a difference in going to result in a difference in these secretion levels or any other activity that maybe is smaller? Does that make sense?
That's a really good question. So our imaging capacity has been limited by the microscope.
Okay.
You know, you buy the microscope you can
Yeah.
afford, and you buy the optics you can afford.
Yeah.
And so that voxel that we've been looking at is kind of our sweet spot for being able to get really clear images to get good characterization. But it is smaller than the scale at which something like angiogenesis occurs. I didn't have time to show all of the data, but that 2020 Lab on a Chip paper goes in and looks at a lot of the differences in those matrix distortions that are generated in just the cell-cell communication ports in the device, where the angiogenic vessel growth has to move through. And we see that it doesn't take a large shift in behaviors. Something on the order of 3 microns of distortion will make vessels grow, but 2.5 microns of distortions won't. So it's a really narrow window. So even though there's a size differential, I think we're still seeing a localized effect due to cellular behavior. That's a really good point, though. I should see if we could, I should make somebody scale that up. Thank you.
Okay, really great talk. I just had a question on the CD44 data. I think it's really interesting. I was wondering, what is the magnitude of strain that you're applying for CD44 to have a protective effect? And then have you guys looked at different magnitudes of strain? And is there like a sweet spot where you do see a protective effect? And are there other ranges where maybe you don't?
Yeah, no, that's a really good point. So everything so far has been kind of capped at like 9 to 10% elongation, and that's because we can get it in both our 2D and our 3D. We have the 3D flex cell plates as well, and so we're trying to match kind of our maximal strain. One of the things that I've asked Miranda to do is go into this magnetic bead assay, and these are much more localized. They're not tissue level strains; they're individual, microstrains. And see if we still see the same effects. But that is a really good point. We've kind of avoided full parameterization sweeps, partially because we're looking more at temporal dynamics as well. So she's got some really cool phospho data that actually shows that like 10 to 30 minutes of strain stimulation alters phosphorylation at CD44 sites. And you start stacking like, okay, 3 time points, multiple strains, multiple frequencies. I also wanted to do static strain, just like pull on it and hold and see what happens, but so far she said no.
Thank you.
How tall are your little fluid cells?
These are 100 microns, so they're pretty thin, even though they're kind of long. And that allows us to really make capillaries rather than having to worry about if the vessels are going to necessarily, stack and be, are they going to develop a basement membrane? Are they going to, all that kind of stuff.
So the idea of shear, when I first got here, we did a study on intimal hyperplasia. So vascular grafts, when you anastomosis, you're around that, you get these little eddies, and I think probably some relationship to that shear stress. The endothelial cells are kind of used to that, and they want that kind of pulsatile flow, and when they don't get it, they actually grow out into the lumen looking for it, and that's where our
yes. And that's
blockages.
a really interesting point, because there's actually differential receptors in different regions of larger vessels. The Tie1 and Tie2 family can sense that eddy current flow and cause differentiation of the endothelial cells. That's why you tend to get plaque buildup at places where that exists. The nice thing about capillaries is if everything goes correctly in your body, they shouldn't need pulsatile flow. It should be fairly continuous flow, because it's supposed to be dampened at that point for nutrient delivery.
How do you make your little cells? What's the process?
So we use traditional soft lithography techniques. We have an entire bench in our lab devoted to PDMS, and I tell people constantly not to touch it with any clothing that they like, because everything's
molds made in clean room, or?
Yeah, we make the molds in the clean room. I designed it in AutoCAD. I'm happy to share it with anybody too. If anybody wants to make these on their own, please let me know. I'll share protocols. I'll share designs.
Molds SU8 silicon, or what are they?
They are SU8 molds. We've had some problems recently getting good fidelity, but the smallest dimension on this is about 20, 25 microns. So Sefu actually is trying to convince me to start 3D printing our molds, and I'm probably going to take him up on that pretty soon. Thank you again. Been a great trip.
Provided in the last few then all change paper.
I realized I wanted to take a picture of you with your like
no.
title slide. I didn't do that.
You
But
make me
then
love
I'll
making.
tweet it,
Okay.
and then we'll get all these
Yeah.
retweets.
Yeah, let's do. Okay, I got to download it again. I'm sorry, I just deleted it.
And then
I'm gonna be famous.
just this. Maybe you can take a picture of me.
I deleted my talk already. Jay was like, we should take a picture. So I'm like,
Yeah, that's nice.
Yeah, I was telling this stuff her. You mentioned your Twitter space.
Oh, yeah.
Bring up social media presence. So she's
I'm the visibility chair
visibility.
for our department.
So like, I guess that's what
That's you.
I guess that's
You're the visibility chair.
Congratulations, you're the faculty responsibility. Hi.
That's a great talk.
Thank you very much.
Thank you for that. I was curious about thinking about the chemistries of these cells. I realized that the tumor environment would be very spatial because mainly, particularly due to shapes and sizes that these tumors are under.
Yeah.
My background, I'm PhD, my background is in a lot of mathematical mechanical.
Okay.
Involves
Yep.
3D modeling tumors, so it has a little bit, I think, just a little bit. And so it made me just think about, it sounded like a lot of those tests are on a tensile sort of pull on these things. Would there be any parallel in a compression sense, or would that be just roughly?
Did you know JoJo?
Oh, a little bit, but
No, there is actually, there's some very interesting work, particularly in the ovarian cancer, about compressive forces inside, because again, that site is fluid built up, it's a lot of extra compression that's not normally present. Joe is studying that in glioblastoma. He's actually got some really cool models that they're developing to add compressive forces to 3D constructs that they're building to look at how that changes brain tumor signaling. Again, like, running the lab is hard, and you kind of have to pick the paths you run down, and so I landed on strain just because of the fibroblasts from some of my previous work. But yeah, no, it's a good question. There's definitely heterogeneous, and it's one of those, like, the calves are contracting, so that's a local strain. They're under compression from the tumor mass growing itself, and there's interstitial flow happening through it because of the leaky vessels, and it's just a mess. I cannot imagine modeling it, which is why I'm hoping to start some collaborations with some modelers who can actually help me imagine.
that would be really interesting. It's our lab is. I didn't personally work on this paper directly, but our lab produced a paper similar working with glioblastoma. It wasn't on mechanical properties, it was about spatial heterogeneity.
Yep.
We looked at the abnormal profile between the core range of these tumors and seeing the differences in their profiles and how that might affect growth. So it was just interesting hearing about the properties of that in ovarian cancer, that there would be parallels with other types of cancer. Do you think that this will become more and more relevant as the sizes of the tumors are larger in the primary? I'm curious because I know it's metastatic tumors, like in, say, the liver, are usually pretty small.
So, yes and no. I love biology. I do. The tumor cells that break off from the primary site, the metastatic cascade, you break off the primary site, you have to intravasate, which means you have to migrate through endothelial cell junctions, get into the vessels. You then have to exist in a state such that you can survive the shear flows induced by being in the vascular system or the left vascular system. Then you have to adhere to the luminal side of that and then extravasate through a cell junction into a secondary location, have to hope that you can adhere to that secondary location, and then, it could be there for 20 years before it starts to proliferate, which is this dormancy question. And so I would imagine, and I have no way to prove this right now, but, you're doing this for another 20 or 30 years,
Yeah.
I would imagine the micromechanical features that make tumor progression worse at the primary site are actually different than what initiate that secondary growth at metastatic site. But I can't prove it.
Yeah.
It's just a hypothesis.
Yeah, but that's really curious.
Yeah. Because, just they have to undergo so many changes to get to that point. So I would feel like they would have some kind of mechanophenotype shift in their behaviors. They also have to re-recruit. If they don't have calf smooth with them, they have to re-recruit some kind of stromal cell.
I wonder if there would be a corresponding adjustment to the microenvironment that they have, or if there's any intrinsic similarity between the primary and the metastatic sites.
We're starting to look at things. So I've mentioned that like brain and bone marrow as a primary site for breast and ovarian cancer. Or we actually have some data that shows if we put one type of matrix in the center, fibrin only, or fibrin plus HA, we can get differential migration events. So cells will preferentially migrate. If they express CD44, they can preferentially migrate towards things that contain HA. Or they're migrating equally, but they're proliferating when they get there. We haven't quite teased out which is happening, but we're trying to work on that. So we're working on that. But yeah, it's a really cool area, and I'm
Definitely sounds like it.
very tired all the time. I don't know if you hear junior faculty coming in here. It's a lot of work.
It does. It sounds really like.
What year PhD are you?
In the spring.
Congratulations. What's the next step? Good. Stay in academia. There's power in the campus.
Absolutely.
Hey, good to see you.
He was asking questions about compression in the tumor microenvironment. And I was like, do you know Dr. Jason?
You're in Herman's lab, right?
Yes.
Yeah, so
He's actually the person I'm going to be reaching out to talk about doing some of this modeling. So.
Yeah,
I'm guessing that's fair.
Herman and I work on a lot of projects together right now. But yeah, so Mackenzie is doing compression effects on malignancy. He's got some interesting, we have a hunter outside our lab. She can watch yeah, so we do a lot of nothing to what Dr. Sewell-Loftin has to that extent, but we have some simple models.
All right. What would you?
I'm just gonna put you in here. Like this. Alright, so let's do. Alright.
And then if you give it to me, I can have
So the question now is, do you want to eat pizza or should we go somewhere?
Is there anything?
It's completely up to you because we could just kick it here and eat pizza too, if that's easier.
I'm okay with pizza. Just want to make sure we get the top.
Yeah, I mean, like, I think the key is that we get you to the airport.
Yeah.
When you
Sorry.
Need to be there, so it's not.
So,
Pizza, we can hang out with Mackenzie and Glenn and stuff.
all right.
I don't know, we're still a department that doesn't like promote academia that much.
Hey, well, I mean, McKinsey did too until I got to work.
I load up a data set and scroll, and then sit there for 2 minutes waiting for it Stand strong.